Hello, here's my presentation on DNA. We're going to be looking at the semi-conservative replication of deoxyribonucleic acid. So in the last DNA and RNA video, we explained the structure of the double helix, the pentose sugar, and the phosphate group. So now we can take a closer look at the interactions within the DNA molecule when replication takes place and discuss the semi-conservative nature of DNA replication. So there's a couple different interactions with water that happen within the DNA molecule. First, we're going to have our positively charged and hydrophobic nucleotides. So these are acidic, slightly acidic, so they carry a positive charge, and they are stacking together tightly to reduce the amount of surface area that comes into contact with the water molecules. Then we have our hydrophilic regions, our negatively charged areas, which is going to be the ribose that has, depending, well, we're talking about DNA, so it'll have 4 OH groups. And then we have the phosphate backbone, which is also negatively charged and therefore hydrophilic. As it happens, DNA replication is an important part of cell division. So we'll look at the process that occurs in eukaryotic cells only today. Replication is semi-conservative, meaning that one strand of the original DNA is preserved for each daughter strand. So you have 2 new strands, 2 new double helixes, 1 old and 1 new in each of the strands. So the 2 helix strands will run antiparallel to each other, so we have a 3 to 5, and then the other strand would be running 5 to 3. Topoisomerases are enzymes with the amino acid tyrosine at their active site. So they're enzymes that carry this amino acid around, and they're only found in DNA. They're involved in preventing supercoiled DNA strands, and they also unravel knotted strands by cleaving the strong phosphodiester backbone bonds. So it can be broken from the 3' or the 5' location, and the broken bonds can reunite once the enzyme is no longer present. Of the ever-confusing Okazaki fragments, they're only found on the lagging strand, so running opposite of the 3 to 5 direction. So the helicase will come up 3 to 5, the primase will come up 3 to 5, and then the polymerase will also come up in the 3 to 5 direction, but it will build in the 5 to 3 direction. So on the opposite strand, that's where you're going to have your Okazaki fragments. So ligase is here. It's another enzyme that's only present in DNA and not in RNA. Its job is to act as a glue that holds the fragments together on the lagging end of the replication fork. So here we have a DNA strand with 2 parent strands. Helicase is entering the scene and is going to break the hydrogen bonds within nucleobases. Really fast, like. DNA primase enters and it's going to prime the bases for the polymerase. And the polymerase follows behind. It's going to build the bases up from the start codon, which is methionine. It's going to continue building the bases in the exact opposite format of the parent strand. So the helicase is gone. The daughter strand is complete. And now you have a final strand, which has 1 daughter strand, 1 parent strand. New DNA strand. Semi-conservative replication.